evaluation worksheet. We do have a quiz today, which is supposed to be like just random questions, randomly chosen. All the lab books should be turned in for grading today. They've already taken half of them, so we'll get the other half into them. The presentations, we are in the Lorenz building on Monday. The Lorenz building. So Lorenz 10 is the 8 a.m. group. Lorenz 36 is the 1:30 p.m. group. Yep. Don't forget that paper is just a regular research paper like you normally do, and that is due by that night at like 11:59, 11:55, whatever. It's Monday, Monday night. So the PowerPoint should be loaded before class. I'll try to remember to send out a reminder to load the PowerPoint, okay? So that needs to be done before class. Like I see these kids out here getting ready for their presentations, and they're making their presentation 5 minutes before class. Don't let that be you. All right. Wednesday, December 6th, our comprehensive final exam. Approximately, remember it's about 130 points, 150 points, somewhere around in there. So approximately 60 of those points, if not 70, will be new material. Mitosis, meiosis, transcription, translation. Okay? I can make a good little test out of that. Yes. Yes. You are risky behavior. Shoving it under the door like that. I mean, you already lost one.
You lost one.
I don't lose it. One of those lab graders has it, or someone took it. Yeah, but at least he shoved it in the door. All right, so those are, and I think everybody did the course eval. I think I tracked everybody down,
All
one way or the other.
tens.
What's that?
All bad.
All bad, I know, but that's okay.
Okay, so for our quizzes, if we have over 100 points,
No, it's only, I only record 100 points for quizzes. So if you have more than 100 points, that's extra credit. Do you understand how that works?
yes, ma'am.
Okay. It's just +8. Okay? I don't look at things minus, I look at things plus. So we just add points on the top. All right, to the worksheet. What question were we on?
13. 11 actually.
11.
Oh, thanks.
Okay, yes. All right.
Are we
Say
turning
hi,
in
you're
these
on
papers?
camera.
Hi. We're going to start off with an exercise that isn't on the worksheet yet.
God, I just left being a mom. I'm a tot. Yes, you can have all these ones.
Oh, okay. You don't like one?
They're
All
okay.
right, write this down.
Favorite?
I like these ones. Apples are my favorite.
Thank you.
We'll see how well you remember last time's activity. Yes.
Hey, same.
I'm calling you guys to come up and fill this in.
Oh no. I'll go.
How many things?
I'm running. At the door.
Anyone else?
Do you have a pick?
Yes, I do.
Can I copy some of your answers?
Which one is that?
Translation.
I didn't do the extension question.
No, it's fine.
He's
So you're talking about how you guys are going hunting tomorrow?
Okay, wait, which worksheet are we
Oh, you
on?
want tonight?
Me and Ken are talking about it.
This is
No,
not
but
a
we're talking about,
worksheet
like,
for
tomorrow
you.
night.
This is an
No,
activity.
I know, but which worksheet are we about to do?
The translation, because Parker's gonna be so loud.
I didn't do this one. I already
Parker,
I did.
Okay, JC's here. She can start us off.
Hey!
What?
That's what's up.
All right, template DNA, complementary DNA, and messenger RNA. Which one is the messenger RNA created from? Which one, template or complementary, is the messenger RNA created from? Template, like the
Boom.
name sounds, right? So, if you're making messenger RNA from the template, we should have the complementary binding of the nucleotides. So TT would bind with AA, A would bind with U, C with G, and G with C. Remember, we know it's messenger RNA because there's a U in there. There's no U's over here. The complementary DNA is going to be similar to the messenger RNA, but it will have U in place of T. Does everybody understand that concept? Okay, so on the next strand, if we know the complementary strand, what is the template strand?
For this one,
What is the template strand for that one?
C, C, G, C, C.
So we have, what would bond normally? Guanine, cytosine, guanine, cytosine. Now what would the messenger RNA look like?
That's where 14.
Math.
It'd look like G, G, C, G, G. There's no T's in there or A's, so we don't have to worry about the uracil, right? Now, what about this one? We're going to go backwards. Reverse transcriptase, so to speak. Here's the messenger RNA. We know messenger and complementary are pretty much the same, right? So this would be A, C, G, T. All right, what is the template then?
Did you really put this theory of
Oh,
F.
there's no A. Okay. Let's do this one more here. It looks like we've got it pretty good. So the complementary here would be A bonds with G with
C, T, G, A, D.
That's hard to see G. Okay. And then this one would be
A, G, A,
C.
C, U.
oh hold on. No. We're this should be the same. Sorry. This should be the same as this, right?
Yeah.
Not binding with this. So this one should be what?
T,
U. C U.
G, A, J.
Okay, there we go. Woo! Because these guys are basically the same. This is what binds with the template.
What are you doing?
So these have been the same except for U's instead of T's.
Oh, so we're not using the complementary DNA, we're going off the template.
Always go off the template with the binding. The complementary is basically the same as messenger RNA because it also has to bind with the opposite strand.
Okay.
Yeah. No, I was like going off there too. So let's do this one again here. So if these are the same, then this would be G A U. And then this would be the same C T G G C T A C. All right. All right, just making sure we're good on that.
Famous.
Famous last words. Now, last time we were talking about the process.
Can you turn off the light?
Turn off the lights.
Thank you.
And we said on these amino acids, or on these ribosomes, the ribosome is going to come and attach to the messenger RNA. And if you take cell biology, you get to learn about that process, what brings it in. Okay? But there are 3 different sites on that ribosome for the amino acids to come into. We have the A site, which is accepting the amino acids, the amino site. We have the amino seal. We have the P site, which is where the peptide bond actually forms, and those are actually going to be binding on that tRNA. Sorry. There's also an E site, which is basically the empty site. It's where the amino acid tRNA leaves. Okay? So we said there's 3 different stages. We have initiation. We're going to start with the AUG. Make sure you know AUG methionine. Okay? That's where the ribosome attaches and it tracks in the tRNAs. We also have the synthesis where we're making the strand. That's called elongation. That's where we're bringing in those peptide bonds between the amino acids. Right? Again, we said that is the primary level of protein structure, just the sequence of the amino acids. So that's why we call it a polypeptide formation. We're making multiple peptide bonds between the amino acids. And finally, we're going to release the ribosome, and we're going to release the protein from the messenger RNA with a stop codon or a nonsense codon, in which the tRNA does not have an amino acid, so that indicates the end. Okay, so initiation, elongation, termination, I believe is what they had in your worksheet. What number was that? Oh, yeah, that was number 7. That was number 7. So if you haven't had that, that was number 7A and B. Now, this is not in the worksheet, and so this will not be on the test. But for those of you going on, I want you to recognize that these ribosomes do have a certain size. All right, and I talked about last time as the S, it says 30S ribosome or 50S ribosome, etc. That S stands for Svedberg unit, which is a sedimentation unit for centrifugation, on how these centrifugate out. Recognize that bacterial ribosomes are different from eukaryotic ribosomes. We can tell the difference, and this is how we can target different drugs against bacteria. Okay, so this is how some antibiotics work. They stop the ribosome from functioning to make proteins. If the bacteria can't make proteins, it shuts that system down. So they do have different sizes of ribosomes, so that's one of the things we can look at. Recognize mitochondria do have a different size of ribosome than the rest of the cell. So the mitochondria can make its own proteins, it has its own DNA, it has its own ribosomes. They are slightly different than the rest of the cell. So there are some differences in there, but when we talk about ribosomes, we are talking about the cytoplasmic ribosomes. The cytoplasmic ribosomes, they can either be free or they can be attached to rough ER, right? The endoplasmic reticulum. That's what makes it rough, is it has these ribosomes attached. Okay. So possible reading frames. If we're looking at the messenger RNA. So there's messenger RNA again. Get used to the term 5' to 3'. All right, we're reading in this direction for messenger RNA to make the amino acids. And we break this up into these codon units of 3 different units. Okay? So here's looking at some different reading frames depending on which way we move. All right, so if this A is cut off in a different reading frame, that would code for a completely different amino acid. So recognize there is a place in which these start, right? An important spot. Remember we talked about on the DNA, it's called the promoter region. And on the messenger RNA, we have the AUG sequence that's supposed to be recognized by the ribosomes to get started reading these frames. So that's how they know where to read. If not, we would have different reading frames. We could end up with different amino acids, which would be the wrong protein for what we need to make. So that's why the AUG is so important. Oh look, it's one of my early animations, my early meth labs, yes. This says formyl methionine. Again, we said bacteria is slightly different. It still starts with AUG, but they have a slightly different coding on there. So what is this funky shape here? What is that? That's a T or a transfer RNA. Remember, it's double. It folds back on itself, and it is charged with methionine. And down here, what's the UAC for? What is that? It's the anticodon, which should match up and bind with the codon. Oh, look, there's the amino acid. Or not the amino acid. There's the ribosome attaching there. So it's going to attach at this very specific sequence to say this is where we start reading. The AUG is the very first codon. This is at the P site. What does the P stand for? Peptidyl, meaning we're going to make a peptide bond with methionine up here. The next codon comes into the A site. Now this one is AAA. It attracts in UUU. UUU codes for lysine. So that is going to come into the A site. My animations are so fabulous, but I think we need more darkness to appreciate them. Oh, look at that. Look at the colors pop here. Look at that. All right, that's initiation. Oh, here's synthesis. I wonder what's going to happen here. That's messenger RNA if you don't know. We're cruising along. Hey, this took a lot of work back in the day. Pay attention. Hey, Oakley.
Ah.
I made this animation about 10 years ago, and I want you to see it. Okay, so notice it's bright colors too, it's supposed to keep your attention. This is cysteine, right? Anticodon, codon, P site. Oh, let's see what's going to happen next. Let's elongate this polypeptide. Oh, looky there. Amino acid, amino acid, amino acid, amino acid. What are these little guys right here in between them?
Leucine.
Peptide bonds. Excellent. And they're at the P site, so we know another peptide bond's going to happen. Here comes that lysine, nope, glycine, sorry, coming in here. And now we're going to form a peptide bond between cysteine and glycine. There it goes, up. Now, little quiz here. On the final exam, there will be, oh, you said it, Austin, functional groups. Functional groups. There are two functional groups up there that are important for peptide bond formation because of dehydration synthesis. Yes, that's on the final exam. I've been talking about it all semester. So what is the very first one up there? C, double bonded O, OH. It is a carboxyl group, all right? Now, what's the other one to the right? NH3.
Methanol.
No, that'd be CH3. This is the amino group. Amino, if it was CH3, it'd be the methyl group. All right, so there is a carboxylic acid on the cysteine and the NH3 on the glycine, and we're going to have dehydration synthesis happen, right? Water given off. Goodbye, water.
Ooh.
And, well, then there were a lot of water is lost, and now we have a permanent peptide bond between what was a carboxylic acid and the amine group. First amino acid, second amino acid. A little review from what we learned early on. And that is a peptide bond. Now we're looking at termination. All right, we have this nice long polypeptide. It has not been folded yet. It is not technically a protein until it folds. This is the primary sequence. This is the primary level of proteins, okay? Over here we have a stop codon that's being coded for, a nonsense codon. There is no amino acid for that, so a release factor releases this polypeptide, and now it can go fold itself.
I totally did. Very well.
And it's going to be released. Now these tRNAs that no longer have amino acids on them, we can get recharged with their amino acids. Can get recharged. And the ribosome disengages. Oh, what fabulous animation. Thank you. Keep the applause down. That's termination. Yes. All right, for prokaryotes, these are the bacteria. They don't have a nucleus. All right? So transcription translation is just coupled. It's just happening right there in the cytoplasm all the time. And you will see multiple ribosomes cruising down the same messenger RNA. They'll just be cruising down the same one, making their peptide sequences. And then once they're released, then they can be folded into a real functional protein. We said transcription translation separated by the nucleus, translations in the cytoplasm or in the rough ER, right, wherever the ribosomes are. If they're in the cytoplasm, it's a free ribosome. But we also here looking at what we call the nascent protein. Notice it starts folding back on itself right away, right, as it's being built. Why does that happen? Amino acids have charges, don't they? They have characteristics. You learned those. Yes, there will be a couple of these on the final exam. I want you to remember those. All right, can you tell if it's charged, uncharged, if it has a special property, okay? So those will be present there. But that's called a nascent protein. It just starts naturally folding back on itself because of those characteristics of the amino acids in that sequence. It'll take a chaperone to further fold it into the functional form. So here's our chaperone. This is again a review from chapter 3, chapter 4. So we take this improperly folded protein that's folded on itself, and here's a molecular chaperone. It looks like a little barrel. It's put in there. It's basically compressed and forced into a proper shape for that protein. Remember, it's all about shape for proteins. If they lose their shape, they're no longer functional. They are called denatured. Denatured. So this is the native form as opposed to the denatured form. And of course, we need ATP to make sure they get properly folded. And that's just supposed to remind you of the whole process. Okay. Now, this has some work that you need to do in your groups. Starting on what? Question 11, I believe. 11 to 16.
Are we doing these questions?
We're doing 11 to 16 right now. Okay. So with your partner, come up with the answers to 11 to 16, and I'll call on you.